Okay, everyone. Thank you so much for being here and welcome to the American Cetacean Society San Francisco Bay Chapter's May 2024 speaker event. I'm Susan Hopp, board member responsible for our speaker program, and with me is fellow board member Wade Cobb. So first, for anyone new to the American Cetacean Society, we are a chapter within a national organization that is the oldest nonprofit dedicated to the protection of whales, dolphins, porpoises, and their habitat. And we do this through education, community engagement, and awarding grants towards cetacean research. We appreciate your donations in support of our mission. Donations fund our research grants to scientists and expenses for these talks. It's easy to donate on our website if you haven't at time of registration. And again, we really appreciate it. So also I wanted to make a quick update on our naturalist class because we are bringing it back and it will be happening in September. Signups will begin in the next few weeks, and we'll make sure that our community is aware of that. It really is a great learning opportunity to hear from experts in the marine mammal and ocean habitat world. So please be on the lookout for that. And now for tonight. So we are recording this session. Please put questions in the Q&A. That is better than the chat. And after the presentation, we'll do our best to get to all the good questions. We're really excited to continue our focus on elephant seals, taking off from the very popular April talk by Dr. Sarah Allen on 40 years of point rays monitoring. In tonight's talk, we shift from land to sea to understand the lives of elephant seals in the ocean. In short, we are exploring how elephant seals make their living in the open ocean. And we are thrilled to have Dr. Patrick Robinson, director of Año Nuevo Reserve, share his expertise and cutting edge research. That's my description from all that I know. So a little bit about Dr. Patrick Robinson. Despite growing up in landlocked Colorado, Patrick developed a love for the ocean and the unique California coastline. This drew him to UC Santa Cruz as an undergraduate in 1999, and he never left. He received a bachelor's degree in marine biology in 2003, worked with Professor Dan Costa to get his PhD in 2009, continued as a postdoctoral researcher, and became the director of Año Nuevo Reserve in 2012. For the last 12 years, he has been facilitating research and education activities at the reserve, helping to train the next diverse generation of researchers and to pursue his primary interest, which is understanding how marine predators make a living in the open ocean. So, Patrick, thank you so much for your work and being with us tonight, and I hand it over to you.
Great. Well, thank you so much for that great introduction, and thanks everyone for joining us tonight. I guess I'll jump right in here and share my screen. So yeah, today we're going to talk about elephant seals at sea behavior and some recent research projects going on. So a little agenda for the evening here. If I can advance the slide there, perfect. So just a brief introduction of Año Nuevo Reserve, just in case you guys haven't been down there. Hopefully everyone here has had a chance to go down and see the elephant seals, but if not, I'll give you just a brief introduction. And then we're going to discuss a little bit of at-sea behavior, some of the current research projects and some of the common questions that I get from folks. And then we'll have some time, hopefully a lot of time for questions that you guys might have. Okay, so for those of you who have not been to Año Nuevo, it's a pretty amazing spot. It's a neat little point. It's a state park and one of the UC natural reserves. I find it super lucky to have a job that brings me here on a daily basis. This is the public trail that you can take as a member of the public and walk out to see the seals. Just as a reminder, if you go during the winter breeding season, you do need a reservation to join a tour, but this is actually a great time to visit right now because you can walk out and see the molting elephant seals on your own schedule. You don't need to join a tour or anything. So Año Nuevo is a state park and one of the University of California natural reserves. We have 41 different sites around the state, and UC Santa Cruz operates 4 of these reserves. And it's just a really amazing network specifically designed for university level research and teaching and public outreach. So I'm doing what I should be doing in my job by sharing this with you guys tonight. We also spend a lot of time training undergraduates, as was mentioned earlier. Train the next generation of researchers is a really important part of our mission so that we can continue to learn more about the ocean environment and help ensure the protection and conservation of all these important species. This is just an example of the undergraduate class that my wife Roxanne and I teach at UC Santa Cruz, where we bring out undergraduates to assist with all the field work, in this case, marking one of our seals with dye marks, which we'll talk a little bit more about later. Año Nuevo is pretty amazing in that we have this small army of docents. I think there's 180 volunteer docents that learn from researchers like myself and the other researchers about all of the new findings, and then share that with up to 100,000 visitors per year. So it's a real megaphone for us to get the word out about these amazing creatures and hopefully inspire other folks to go into similar fields. Before I jump into the detailed research, I just wanted to mention that I am just one cog in the wheel at Año Nuevo. And basically the elephant seal program exists because there has been a series of professors at UC Santa Cruz that initiated and continued the work that goes on there. So I want to be sure to give respect to all of these people. So Professor Bernie LeBoeuf was the one who started the elephant seal research program back in the late 60s when elephant seals first started appearing at Año Nuevo Island. Then he had a wonderful career publishing many great papers. And then Professor Dan Costa joined and took over the elephant seal research program. And now Professor Roxanne Beltran and Professor Dan Costa are co-leading the program. So lots of amazing research, but it's basically because of these individuals that all of that is possible. So during your last Zoom session, I think you heard a bit about the on-land behavior of these guys, but it's so amazing that I had to show a couple of videos here just to reiterate how amazing it is that these guys basically show us so much of their life history through on the beaches here, and we can just observe that directly. So Professor Bernie LeBoeuf did the majority of his research just based on observations on land, which is just amazing for a marine mammal to be able to see all of this firsthand and publish some amazing work based on that. So obviously the males fighting are very iconic, but I'm actually more interested in the female behavior. This is what it looks like during the middle of the breeding season. See all the females with their pups and an occasional male mixed in there. This is, for me, what's more interesting, the reproduction, because from a population dynamics perspective, this is what matters more. You can have fewer males around, that's not a big deal, but if you have fewer females or pups around, that's a big deal for the next generation. And specifically, the energy gain associated with pup production. So obviously the birth mass of the pup is very important, but also that nursing. So these guys have a fairly abbreviated nursing period of just about 27 days, so a little bit less than a month. And in that amount of time, they go from this small individual, 30, maybe 35 kilograms, all the way up to an average of 120 kilograms. So these blubber balls are basically packed with all the resources they need to survive the first few months of their life when their mothers basically abandon them on the beach at the end of the breeding season, and that's the end of parental care. So it's a really neat system in that all of those resources are passed to the pup that then becomes a weanling, and it's very easy to measure that just through the weight, the mass gain of these animals. So we can measure the mass of weanlings and understand something about how well the females did on their past foraging trip and how likely they are to survive in future years. One of the ways that we keep track of all of this, and I'm sure Sarah Allen covered some of this work up at Point Reyes, is through our demographic program. So every year we put flipper tags in about 500 weanlings. And so this is an example of one, just uniquely numbered green flipper tags. All of our flipper tags on Nuevo are green. So if you ever see a flipper tag that's green on a seal, you know it's from Nuevo. And then we do this year after year and then basically try to find those animals again. We have research teams going around on almost a daily basis, looking through our dense colony here to find those green flipper tags. And it can be pretty challenging at times. This is what it looks like sometimes out there at Nuevo. I don't know about you, but I'm not seeing very many green flipper tags in that. So it's pretty challenging. So we spend a lot of time looking at these guys through binoculars and cameras, but it's still pretty challenging to read those flipper tags. So we do have one trick up our sleeves. We have dye marks. So once we find a seal that has a flipper tag, we can use our Clairol hair dye, available at your local Safeway, and use our little stamping tool and stamp the number right on the side of the seal, very non-invasive way to mark an animal. And then that will stay on the seal until its next annual molt. And that enables it to be able to be seen by us and by other people and record information about that animal upon each observation. So here's just a small example of the observations from one particular seal, and we've tracked thousands and thousands of seals this way. So each row corresponds to one observation, and we have the tag that was observed, the mark that was observed, the location, the date, whether it had a pup or not, the molt status, if there's something interesting done to it, like attaching a time or a satellite tag. Sometimes we replace tags if they need to be replaced. Anything interesting, maybe it has a shark bite. It's all recorded in this database, and then we can go and mine that for all sorts of useful information. So one of the many things that we can do is when you amass all these data across many decades, we can ask some pretty amazing questions. And this is from a paper that Dr. Bern Leboeuf wrote a few years ago that basically utilized several decades of these observations. And it turns out that you can look at the mother's age and the pup mass at weaning to basically infer how good of a mother a particular age seal is at producing a pup that is likely to survive into the future. So we can see here that a mother's age matters a lot, especially during the first few years of reproduction. The 3, 4, and 5-year-olds have smaller pups than the older individuals, and that affects their future survival. And likewise, we can look at the total lifetime reproductive success of an adult female. So this takes a long time to get these data. So we have to observe individual animals for up to 20 years, and then do that for many, many individuals in order to get a plot like this. So in short, each bar represents the lifetime reproductive success of particular seals. So most seals have 0 pups in their life. So most adult females don't actually reproduce. So in fact, these black bars indicate that like 83% of females that are born this year will never survive to reproduce, which is a pretty amazing statistic. But then if we look at the 1 there, so how many have 1 pup? About 6%. And it gets a little, it gets progressively worse here, but the number that have 2 in their life is about 3%. The number that have 3 is about 1.5%. So just a very small proportion of animals are having a disproportionate impact on the next generation. And we have examples of some females that have more than 10 pups in their lifetime. And so these we're called super moms because they're just so good at producing pups to help that next generation. So these are the types of things that we've studied on land, but it's been noticed for a long time that a seal, when it is about to depart, is quite skinny. It's depleted of all these resources, either from giving birth to a pup and nursing it for a month, or staying on land for a month while molting, which is also fairly energy intensive. So when they are about to head out to sea, they look very skinny like this, and then they go off on these epic foraging migrations and come back, and this is what they look like. It's kind of a fun little animation there. I'll do it again. So they just put on this immense layer of fat in order to survive that long period of time on land without eating or drinking anything. And a lot of the research that Professor Dan Costa and Professor Roxanne Beltran and research that I've helped with has addressed in the past couple of decades is how these elephant seals make a living out at sea in order to do all these amazing behaviors on land. So it sounds kind of simple, but basically, how do they gain all this mass? What strategies do they use? What adaptations do they have? These are amazing animals, and it's very difficult to study them when they're out in the open ocean. Very easy on land, but much more difficult out in the open ocean. And part of that is because they really do go far. I mean, they're out in the middle of the Northeast Pacific. So, like, this is what it's like to be an elephant seal. Like, you just look all around, and this is where they spend most of their time. Except this actually isn't even perfectly accurate because they're not spending that much time on the surface. Most of their time is actually spent well below the surface, and in fact, well below the photic zone, so in complete pitch black, dark waters. So the single place where they spend most of their life is hundreds of meters down in the middle of the Northeast Pacific without any sort of cues or light. It's an amazing thing that they can survive in that environment and forage successfully in order to come back and reproduce. So how do we study them? So if I were to try to follow an elephant seal, with a boat or on scuba, anything like that, I would probably be able to follow them for about 30 seconds, and then it would just disappear into the abyss. So we really are reliant upon biologging. So this is the attachment of electronic instruments to study the behavior of animals. And so this is a burgeoning field. When I was in graduate school, I went to the second ever biologging science symposium, and it was just a fledgling field at that point. And now there are hundreds of researchers around the world that are instrumenting marine animals, terrestrial animals, birds, insects, just a huge number of animals. And this is a giant field now, and it's really established itself. But one thing that's really neat is that a lot of the work done on seals, in particular elephant seals, was sort of pioneering in this field because of the ease with which we can attach instruments and recover them fairly reliably. So researchers, if they're interested in studying tuna, they might not want to put out their precious, very expensive new prototype tag on a tuna and just have it be lost into the abyss and never return to deliver data. If they want to test that first, they can work with us to instrument an elephant seal and test their tag, collect some data, refine it, and then finally deploy it on their species. So that's one of the amazing things about elephant seals, is that we can use them as a platform to enable the study of other seals and other species as well. Okay, so what's an example of some of the biologging that we do? The biggest one that we do is tracking and diving. So we now have tags that can kind of do it all. So we just need to put one tag on an animal, and it can serve as a tracking tag, a diving tag, even an oceanography tag. So this particular one that is in the photo here is a sea mammal research unit conductivity temperature depth tag. It's a satellite relay data logger. It's a mouthful, but it basically is transmitting to the Argos satellite network, and that helps us get the position of the animal several times per day, as well as transmit data in near real time. It also has a time depth recorder built into it, so it's measuring the depth that the animal is reaching every few seconds. And then it also has a conductivity cell on it, and that might be a weird thing to measure. So conductivity and temperature, from those two measurements we can get salinity, and temperature salinity plots are something that oceanographers are very interested in when they're studying the open ocean, studying different water masses. It's basically the fingerprint that allows us to identify different water masses. So you can think of that as kind of like the habitat where the animals are in. So each of these tags can record a variety of parameters. No one tag records everything, so we get to pick and choose depending on the particular study. In the next photo here, we have a GPS tag. This one was done because we wanted very fine scale resolution on the movement patterns of the animal. And those Argos satellite transmitters are great because they're actively transmitting, so we know where the animal is, but the error on those is pretty high. like upwards of 10, maybe 15 kilometers in some cases. So, that's like halfway across the Monterey Bay as just an error, so that's not great. So if we want to know the exact location the animal surfaces after every dive, we can put a GPS tag on them and get that information. But that comes at the cost of not putting out some other sensors. So again, we get to pick and choose depending on the project. Okay, so what have we learned from some of these tags? So we'll start off with the satellite tracking tags, the Argos tags. So this is kind of a fun little sequence here. I'll start off by showing you what was known about elephant seal distribution patterns in the early 1990s. Basically, we just knew that they existed along the West Coast, but didn't really know where they went beyond that. And then Professor Bernie LeBoeuf in the late 90s published a paper in which he put out about 50 time-depth recorders and satellite tags on elephant seals. The red there are the males and the yellow are females. This is back when the Argos satellite network wasn't as big as it is now, so the tracks are very kind of wiggly, not very straight lines there, but you still get the idea that the seals are going very far away from the coast. And then the baton was passed off to Dan Costa, and he started the Tagging of Pacific Predators project, and that was right when I joined his lab as a graduate student, and that was very fortuitous just because he had a large amount of funding coming in for that project. So I was one of the graduate students that went out and instrumented lots of these seals. And we were able to collect a huge amount of data, tracking dozens and dozens of adult females in order to really fill in the gaps here. So we had an idea about what the females were doing, but once we were able to tag a much larger number of them across many years, we can begin to ask some really big questions about what animals are doing, what's normal, what's abnormal, different ages, what's the effective age, what's the effective size, like lots of different questions once you have a large enough sample size. So that was a pretty exciting thing. And the next slide here is one that shows the animation. So the still images are great, but what does it actually look like when the seals are moving around? So obviously this is highly sped up, so bear with me here, but this is a year in the life of the elephant seals of Nuevo, and this is just adult females, I should mention. So we'll start off with the seals departing from the breeding season on their shorter post-breeding migration, so taking off there. They're gone for about 72 days and then just kind of out and back, come back in for the molt, spend about a month on land to molt, and then they're off again on the post-molting migration. This is the longer trip, about 240 days. And then they're growing their fetus, and then they come back and give birth to a single pup during the breeding season, usually mid-January. So that was obviously very sped up, but that's basically a year in the life of the colony. So you can see there's not very much of the time they're on land. They're actually out at sea for quite a while. Okay, so some basic movement stats that we've learned from those satellite tracking tags. I'll split these data into two categories here, the breeding trip and the post-molting trip, because they each have 2 trips per year. So the post-breeding trip, we have a duration of about 75 days, an average maximum distance of over 2,000 kilometers. So the average individual is going 2,000 kilometers away from Nuevo. And the total distance, if you take into consideration all of their like little wiggles and movements, is nearly 5,000 kilometers. So these guys are absolute athletes. That is a tremendously huge distance to move over the course of just 70 days. And then the post-molting migration, a little bit longer, obviously, 218 days, and that allows them to get quite a bit farther away from Nuevo, on average 3,200 kilometers, and nearly 10,000 kilometers of swimming. So when we take all this in consideration, they're swimming about 50 kilometers per day while they're out at sea. So I don't know about you guys, but I'm lucky if I can swim 1 kilometer, and that does it for me. So these guys are just amazing athletes. And that's not taking into consideration the vertical component. This is just the horizontal component. Okay, so why do the elephant seals go so far out to sea? I always like to compare California sea lions, which are also at Nuevo, with elephant seals, because you might think that they do something similar because they're both pinnipeds, they both reproduce at Nuevo, they both haul out there, but they're doing such different things. So California sea lions are exploiting the area right around the coast, and elephant seals are going these vast distances away And it just begs the question, why are they going so far away? So this is where the oceanography comes in. So just generally speaking, this is going to be a little bit of an oversimplification, but there's basically two gyres, the subarctic gyre and the subtropical gyre, and the gyre boundary in between them. And generally speaking, that is just a very productive environment. So you might hear people call that the transition zone. There's also a chlorophyll front in there that is marking a productivity delineation there. So just lots of interesting things going on at that gyre boundary. Now, this is just kind of like a stylized diagram that I made, but let's look at some real data. So this is a kernel density distribution plot of all of our tracking data, and basically the red corresponds to areas where seals are spending more time, and the greens and blues are the areas where the seals visited but didn't spend very much time. So you can see that most of the red is right along that gyre boundary as measured by sea surface height anomaly measurements from satellites. So it's just kind of neat to be able to use oceanographic products that are available to us to understand where the seals are going. And so they're probably up there searching around for these areas, and we probably could predict where they're going with pretty good accuracy based on these satellite measurements. Okay, so that's the movement, and then diving behavior. So each of the tags that we have records depth in addition to the location of the animal. This little animation I have here is highly sped up, so each of these dives is more than 20 minutes in duration. Oops, get back here. So this is the average kind of the day in the life of an elephant seal. It's my joke. It's the 9 to 5 work day for an elephant seal. So in any given day, they might do several dozen dives. And this is a very typical kind of nighttime to dawn to daytime pattern here. So in general, they dive a little bit shallower during the nighttime, and then have a series of what we call drift dives right at about dawn, and then switch over to doing more deep foraging dives during the day. So essentially, they're shifting their depth based on how much light is available. They're going well below the photic zone, but we think that the prey that they're going after are moving in function of the ambient light levels. So that's just sort of an interesting thing. So with every seal, we get like literally tens of thousands of dives like this, and we've instrumented hundreds of animals. So it's just an amazing amount of data, and it's very rich. So we can go through these and learn a lot about what the animals are doing just based on this diving behavior. And when we pair that with the movement data, we get a really good picture about what's going on out there. Okay, so some basic diving statistics, again, for the post-breeding trip. This is the shorter trip. We have an average of 4,500 dives, average depth of 527 meters, a total vertical distance of 2,400 kilometers. So this is on top of the other distances that I was talking about, because those were just horizontal distances. Now we're talking about vertical distances with diving. So just amazing, amazing distances. The duration on average is about 22 minutes, and they just need a little over 2 minutes to recover from that. Then on the post-molting migration, a larger number of dives, obviously, more than 11,000. A little bit shallower on the depth, interestingly. Total vertical distance, almost 6,000 kilometers. It's pretty wild. A little bit longer in the duration, 24.5 minutes, and similar recovery time, 2.5 minutes. All of these values, I should point out, are just averages. So these are astonishing just as averages, to know that the average dive depth of a seal is 527 meters. It blows me away every time, because when I think about like my own diving ability, if I take a breath hold dive, I'm lucky if I can get down to like 10 meters. I'm a scuba diver, so I go free diving occasionally, and I think the deepest I've ever gone on a single breath hold is about 10 meters. And so the idea of going to 527 meters is just absolutely wild. So these are just the averages, but the maximum depth that we've been able to observe is 1,756 meters, which is a little deeper than a mile, and a maximum duration of just shy of 2 hours. So the duration, I think we can all conceptualize, because, okay, that's about the length of a movie. So sit down, watch a movie on Netflix, try to hold your breath for the entire thing. That's what the elephant seals are doing. The maximum depth is a little bit harder to wrap your head around because we don't do vertical motions like that very often. So I like to put that into perspective of some of the world's tallest buildings. So this is as of like, I think 10 or 15 years ago, but these, some of the tallest buildings in the world. And you can see that the tallest one, is approaching 900 or 1,000 meters, something like that. So if we shrink these down, the deepest dive that an elephant seal has done is about there. So I like to think about going to one of these buildings, getting on the stairs and like walking all the way up while holding my breath all the way up to the top, having lunch up there, and then walking all the way back down, and then taking your next breath. And that would just be a walk in the park for an elephant seal, nowhere near their maximum. So these guys are just absolutely amazing. And they do this routinely. So they're diving continuously when they're out at sea. And there's about 23 minutes down, 2.5 minutes to the surface for months on end, day and night. Okay, what is the perspective here? So is this impressive in comparison to other marine mammals? So if you look at the plot on the left there, you see the top 5 animals there are other pinnipeds. And you can see that elephant seals are by far the deepest diving of the pinnipeds. So both northern and southern elephant seals are pretty similar, and they're diving way deeper. So that's pretty impressive. They're not the deepest diving marine mammals, so there are a few different species of whales that can dive deeper. But those guys have the advantage of having a larger size. So in the plot on the right, you can see that elephant seals are a big outlier. So that's the point there in the middle that has the arrow by it. So when we look at the body size of an animal, so along the x-axis there, and then compare that to the maximum dive depth, in general, the larger body size you have, the larger your maximum dive depth can be. But elephant seals are an outlier there. Like they should not be able to dive as deep as they do. So they've really mastered that. And so there's probably a good reason they're going after prey resources and evolved over millennia to do that. But it's just important to point out that they're doing something very different than many other pinnipeds. And really, in many ways, they're probably behaviorally a little bit more like a beaked whale than they are like other pinnipeds. Okay, so what are they doing out there? So we can attach cameras to animals. This is work by some of our Japanese collaborators, Yasuhiko Naito and Akinori Takahashi. They basically developed some instruments working with a Japanese tag manufacturer called Little Leonardo, and we were very interested in what the animals were eating. It's very hard to know from some of this time depth recorder data exactly what's going on. We can look at the depth wiggles and try to figure out in a particular area of the track or the dive that they might be eating, but knowing what they're eating is very challenging. So they developed a small camera that we could attach to the top of the head of the seal, and would basically, it's very much like a critter cam from National Geographic, if you guys have heard of those, except much smaller, and it is triggered by a depth and a time delay and an acceleration trigger. So it basically just takes little snippets of video right at the right moment to capture some of these things. And so they got some amazing results back from these cameras, a whole bunch of different fish and squid species. But the bottom line is that the majority of the prey items are myctophid fish. So these are lantern fishes, like in the upper left there with the A. So basically like small little fish. So they're going after just dozens and dozens of these small fish, and then also taking advantage of other foraging opportunities as they present themselves. One of the things that we can also do is attach a little jaw acceleration logger. So in addition to a camera, we can put a little device on the chin of the animal, and that records, just like your Fitbit or smartwatch will record every step that you take. These record every jaw opening event that an animal has. And so we can plot that on a time depth recorder record to see where they are feeding. And so we can validate all of this with those videos. Those cameras are something that they're very expensive and customized, so we can't put them out on every animal. But these jaw acceleration loggers are a little easier to put out. And then we can also validate just the shapes of these dives. So you can see the dives that have little wiggles at the bottom have more foraging events. And then those non-jaw motion event dives in the middle there, those are the resting dives, don't have any foraging events. So that met our expectations there. And now we can guess basically that a lot of our dives that have lots of wiggles at the bottom are probably foraging dives. Okay, so now that we know what they're eating, how do they actually capture prey in the open ocean, hundreds of meters down where there's no light? So this was a big question that our Japanese collaborators had. And I give them a lot of credit for this because they had these questions very early on and worked with tag developers to develop custom instruments to address these specific questions, and then worked with us to deploy these instruments over the past 15 to 20 years. So in short, elephant seals are feeding in the deep, dark ocean. They don't have active biosonar like many whales do. They have very sensitive eyes. So one of the things that you notice when you look at an elephant seal is just it has very large eyes. Its pupils are very, very, very tiny on land. I mean, just pinholes, even when it's pretty dark. So you can surmise that they're very sensitive eyes. And we also know that they have a very sensitive whisker bed, so they can probably detect little microturbulent structures in the water with their whiskers. So when you don't have access to light, probably they were using their whiskers, but there's also bioluminescence with the prey items. So those myctophid fish and other species have photophores along them that can light up. So it wasn't exactly clear to anyone what the proportion was of these two methods for finding prey. Are they just finding prey that are bioluminescing, or are they able to detect prey that have no light emissions of their own? So our collaborators worked to develop some tags that would help address this. And one of the ways they can do that is by putting an even smaller, tiny, tiny little video recorder on the cheek of an animal. And so that's what this guy is here. So again, this is on the cheek of the seal, and you can see the whiskers, and you can see them move forward a little bit, and you're right there. So it's a little obvious when they do it because the base of the whisker kind of glows a whiter color there. So you can see when they protract their whiskers, and they've deemed this rhythmic whisking. So it requires a muscle contraction for them to push those whiskers out. And I think it's very similar to a cat. So when a cat is exploring its environment, it'll project its whiskers out. And I think the seals are doing something very similar to that, especially when they reach a certain depth. Then they put their whiskers out, and they seem to be able to detect even tiny, tiny little vibrations of a single prey item that may have swum by like 30 seconds earlier. So a little fish swims by, and that leaves a little microturbulence trail that takes a while to dissipate. And the seals will go through that, sense it with their whiskers, orient to it, and go after and catch that prey item. And then there's also the bioluminescent prey. So when we look at all of this, we can know the diving behavior of the animal. We have the jaw acceleration to know if the prey item was likely captured. We have the whisker video to look when they were searching for prey. And you saw that little blue item there. So that was an example of a bioluminescent prey item. So from this, we can actually look at the proportion of their prey items that are bioluminescing just before they're captured. So here's an example of a time depth recorder record from a single dive. Those black squares correspond to individual feeding signals. And then the irradiance is the amount of light that the tag detected, so basically bioluminescence. So you see that there was one bioluminescent event with that red circle, but many feeding signals. So that was interesting, but they wanted to analyze a lot more data than that. So they accumulated data across many seals, many dives. And basically, the percentage of bioluminescence with feeding signals was very high. So if the tag detected bioluminescence, the seals were successful at capturing the prey item, usually. But the percentage of feeding signals that had bioluminescence was pretty small. So there were a lot of feeding events that did not co-occur with bioluminescent events. So this told us that if a fish or squid bioluminesces, it's basically caught. Like the elephant seal can find it very easily and capture it. But the seal, most of the time, is not using that. So they're able to use their whiskers to capture most of the, or to detect most of the eventual prey captures. And then the final bit, actually consuming them, another graduate student in Dan Costa's lab showed that they are suction feeders. So they'll get very close and just suction in those fish or squid. So this gives us a very detailed picture about what the animals are doing and what it's like to be down in the deep, dark, open ocean foraging. And when we first started studying these animals, we just really didn't have any idea that whiskers were quite as important as they are. But it turns out that those whiskers are absolutely essential for their survival. Okay, so then moving on here to some other things we can do with the time depth recorders. So here's our kind of day in the life of the elephant seal. So it turns out we can, as I mentioned before, 8 different types of dives here. And let's zoom in. Basically, so we take the raw time depth recorder data, convert it using some custom software, and then we can identify different shapes of dives. And so these are very stylistic diagrams here, just kind of showing the rough general shapes of them. But the first 2 are transit dives, the C dives are drift dives, D dives are foraging dives, and E dives are benthic dives. And for a long period of time, we were most interested in the D dives, the foraging dives, for obvious reasons. We're very interested in the collection of resources to bring back to have their pup and nurse that pup very well. So it was the amount of foraging that mattered the most. But it turns out that C dives are actually quite interesting as well. So let's take a closer look at those. So this is a stylized dive. Let's take a look at a real dive. So this is like real time depth recorder data. So the animal spent a little bit of time at the surface, actively swam down to 250 meters, and then started the drift phase of this drift dive, and then drifted down to about 450 meters, and then actively swam back up to the surface. So there's an interesting thing that researchers before my time noticed, was that as you look at these dives, so these drift dives typically occur in the morning, early morning hours. So, and they occur on most days throughout the migration. So early in the migration, that angle there was a little bit steeper. And early in the migration was the animals are kind of skinny. And then later in the migration, that angle might actually go positive. So they might actually float upwards during that phase. And that's when the seals are a little bit fatter, a little chunkier. And then some additional research, especially just recently by a graduate student in Dan Costa's lab, Jesse Kendall-Barr, documented that basically the animals are sleeping during this drift phase of the dive. We knew that they were resting, but she was able to prove that they're actually sleeping, which is pretty neat. So what's going on here? So again, some of our Japanese collaborators came out. Next one is by researcher Yoko Mitani. She put out an acceleration data logger that could record the animal's 3-dimensional movement path. And this is just that drift phase. And this earlier diagram makes it look like a straight line, but in reality, it's kind of a corkscrew. So we think that they fall asleep, flip onto their back, and slowly spiral down several hundred meters over the course of about 10 minutes, and then wake up and swim back up to the surface. Now, this is a very weird behavior. It sort of makes sense, though. I mean, they have to sleep at this point. Jesse Kendall-Barr showed that they're actually one of the mammals with the lowest amount of sleep required, at least while they're out at sea. They make up for a little bit while they're on land. But so this is super interesting. So they're not actively propulsing themselves during this whole 10-minute period, which means that the rate at which they drift down through the water column represents their buoyancy. So I mentioned that the skinny animals have that steeper slope and the fatter animals might have a shallower slope or even a positive slope. So we can actually track their foraging success based on these drift dives, how quickly they fall through the water column. So this next plot is looking at basically how that drift rate changes throughout the migration. So this is the long trip from June to January, and you can see that it starts off negative. So the animal was losing condition initially, so that drift rate was getting steeper and steeper, but then started successfully foraging, and then it was in positive energy balance throughout most of that migration, and then looks like had a not very successful trip home, just was kind of like breaking even most of the time. So this is amazing, right? We can look at on a day-to-day basis how successful the animal is. And we knew from the time depth recorder data that they were feeding, but knowing how much they're feeding and if that is meeting or exceeding their energetic needs is pretty valuable and very rare amongst marine mammals. So we can take these data and basically map them onto the actual track of the animal to see where the animal was having foraging success. So this is actually an animal that I instrumented down in Mexico in 2005. And you can see that it went up the coast and then out into the middle of the Northeast Pacific and then back a different trajectory and then kind of followed the coast back down. But most of its foraging success happened in that area off to the left there. So most of the red dots are over there. So this is just one animal, super interesting. But what's even more interesting is that we can take a whole bunch of tracks and then run that same analysis and see those important areas for the entire population. And sure enough, that matches very well with where the animals are spending most of their time in that gyre boundary. So just to remind you, right there in that middle area is that gyre boundary. So the animals are seeking out that area and spending lots of time there and having foraging success there. So it's lovely when these stories make sense, right? They're not spending time in poor foraging areas, they're spending time in the areas that are most productive for them. Interestingly, in this image, you will see that a couple of animals go all the way up to the Aleutian Islands. And that is a behavior we typically associate with males. Males need to gain a lot of mass very quickly, so it's probably very productive up there, but it's also probably more risky. But these females went up, and as you can see, were very successful up there. So we see some females behaving like males and going up and feeding in these coastal areas, but we haven't tagged very many males to date, but I'm not aware of a single example of a male feeding out in the open ocean like most of the females do. So it's sort of an interesting difference between the genders there. Okay, then moving on to some more tracking information here. So this is something that I looked at with some tracking data that I collected for my PhD dissertation. And then Professor Beltran kind of followed up looking at this as well. So elephant seals are amazing navigators. So I love this slide because it shows you 3 different types of interesting navigational feats that the animal is capable of. So there are 3 different animals in this plot. So we'll start with number 3. So this animal took off from the San Benito Islands down in Mexico on its short migration, and then went off to sea, and then turned around and came back and pretty much followed its own path directly back. Now that sounds like very easy to do, but the scale of this is 1,500 kilometers, or nearly 2,000 kilometers. And so to maintain a very straight trajectory out and back and have those match over that sort of time span across that distance is pretty amazing. And if you just set me off even on land in a straight line and said, go in a straight line for 3 weeks, I would probably go in many loops over that period of time. I would not be able to navigate very efficiently without a GPS or something. So that alone is pretty amazing. Then let's switch up to number 1 there. So animal number 1 goes straight out to the Aleutian Islands, hangs out up there. So again, that nice straight section is pretty interesting. Then on its return migration, starts heading back along the same path, but then takes a left turn and heads up to the Alaska gyre and feeds up there for a while, and then doesn't need to get back onto its original track. It knows that it needs to reorient itself to go back home in the most direct way possible. So that's pretty interesting. So that is a little bit of evidence showing that the animal kind of knows the direction of home at all times. Then animal number 2 there, you'll see that those are 3 different migrations, all during the post-breeding trip, but of different years. So same animal, but different years. And you can see that it does this out and back. It has the flexibility to go anywhere in the Northeast Pacific, and it goes pretty much to the same areas along the same route, and all 6 times it passes that center point, it goes over that very small geographic area. So I think with some further research, there might be some interesting information that can be gained from that point in the middle. Like, I want to know what's going on at that magic spot where all 6 paths are crossing. So from this, we know that elephant seals are pretty amazing navigators. And then Professor Beltran took this a step further by looking at their map sense in a quantitative way. So what she and one of her students did was looked at the point at which the animals turn around from their migrations, and then looked at the date with which that happens and how far away they are. That sounds like a weird thing to do, but basically it's answering the question, do the animals know how far away from the colony they are and how long it takes to get back? If they just go off in a certain direction and then come back and don't know their timing, that would indicate that they don't really know where they are necessarily. But if they can time things well, particularly for the breeding season, they're giving birth to a pup, they need to arrive on time, otherwise they risk giving birth in the water, which would be lethal for the pup. So they need to be able to get back in a very timely fashion, but yet maximize their time in the foraging area. So turns out when you look at all those turnaround dates, the turnaround date correlates very well with the turnaround distance. So the animals that are farther away turn around earlier in order to get home earlier. In order to get home at the same date. So they're all trying to get back to have their pup, and the ones that are farther away know that they are farther away. So this sounds like very simple, but it's actually really difficult to show that animals have a map sense. And this was able to document that the seals basically know where they are when they're foraging out at sea and how to get back and how long it'll take them to get back, which is pretty amazing. Okay, so next we'll switch over just to basically a few questions that I typically get, just to head off some of those before our Q&A period. So I like to talk about some of the adaptations that allow the elephant seals to do some of the amazing things that they can do. And we don't have all the answers by any means, but we know some of the tricks that they have up their sleeve. So one of them is decompression sickness. So a lot of people, when they hear about these amazing dives the animals are capable of, they ask, well, why don't they get the bends? Like human scuba divers will get the bends if they dive to these depths for these durations. And the main reason there is that they're not breathing pressurized air. So they don't build up nitrogen in the same way that we would. So basically their lungs collapse at a fairly shallow depth. Elephant seals actually exhale before each dive, which sounds pretty wild. So most of their oxygen is stored in their blood. So they actually collapse their lungs probably at about 50 meters or even shallower. And so there's no air exchange, there's no nitrogen exchange into the bloodstream. So basically nitrogen can't build up in the same way that it can with human scuba divers. So they've got that one covered. Okay, so then they also have a mammalian dive response, which many mammals do, including humans. So basically, if you submerge your body, particularly your face, your heart rate slows, you have peripheral vasoconstriction, so blood goes away from your extremities. And these guys, they have splenic contraction, so their spleen, which is filled with oxygen-rich blood, slowly contracts and basically delivers additional oxygen to them. And then they have a very slow metabolic rate, so reduced oxygen use. So all of those things happen just kind of naturally without them trying as a function of their dive reflex. Okay, I mentioned that the elephant seals exhale before they dive. So how are they able to do that? Well, the seals, this is just a general diagram comparing different species here, but if you look in the upper right, you'll see that particular one is a Waddell seal, but elephant seals are pretty similar. Phocids in general store most of their oxygen in their blood. So very little in their lung, and a decent amount in their muscles, certainly way more than us, but the vast majority of their oxygen storage capabilities in their blood. And they have a few different tricks that allow them to store lots of oxygen in their blood. And one of them is just having a lot of it. So these guys have, I forget the exact number, but something like 60 liters of blood. I mean, it's just an insane amount of blood. It's their single largest tissue. And so having a lot of blood, they have a lot of red blood cells, so their hematocrit levels are very high, and they pack more hemoglobin into every red blood cell than humans or many other mammals do. So basically everything about their blood is very well equipped to carry lots and lots of oxygen and deliver that to the tissues. We did have one researcher, Dr. Jessica Meir. Fun fact, she's now an astronaut, but she did her PhD in part on elephant seals, also emperor penguins. So just like to highlight the fact that she's gone on to some pretty amazing things in her career, and elephant seals helped propel her with that. While she was a graduate student, she was working out of UC San Diego, but doing work on Nuevo. And she worked with a group that had developed an instrument that was recording the partial pressure of oxygen. So it's going to be oxygen saturation of the blood during free-ranging diving activity. And she was able to show that the oxygen saturation of seal blood goes down to like 5% in some cases. I always like to point out that humans lose consciousness well above that. So like literally if you were to experience what the elephant seals are experiencing on every dive, you would pass out. So these guys have some pretty amazing adaptations to be able to deal with that. I already mentioned the lung collapse, but just the different structures of the lungs in different pinnipeds allow them to do this. So in particular, the cartilaginous areas that normally hold our airways open, there's a lot less cartilage in these guys. So that enables the tissue to collapse upon itself and to eliminate the air exchange at that alveolar interface. Thermal balance is a big thing. So this one's pretty obvious, but something like a sea otter relies upon fur for warmth, so a trapped layer of air. Deep diving animals cannot do that because the pressure would shrink that air layer down and negate any thermal benefits. So it's kind of like wearing a wetsuit. If you're a furry animal, you can't go that deep without getting cold. So elephant seals use an incompressible tissue, so blubber. So you can see here in this kind of horrible cross section. This is not done by anyone I know. This is like a textbook from decades ago, but it very clearly shows the amazing cross-sectional ring that is the blubber layer. So just an amazing ability to thermoregulate based on that. And they do have the ability to use thermal windows as well to sort of send out blood to their flippers and their periphery to dump heat when needed. These guys also have an amazing ability to conserve water. So they can concentrate their urine. They have very low respiratory evaporative water loss, in part because of their nasal turbinate structures, so very complex structures in their noses that capture water. And then they also produce water. Sounds kind of crazy, but whenever you burn fat, you actually produce water. It's a function of glycolysis. So you and I do this too, but they're burning mostly fat and can conserve the water that they produce very well. So it turns out if you burn 1 kilogram of fat, you get 1.1 kilograms of water. So that's probably why these guys don't need to drink any water while they're out at sea. Okay, that wraps it up for me. Looks like we're right at about an hour into this. So I'd be happy to take any questions you guys have. And it's a lot to cover. There's certainly hundreds of scientific papers that I didn't address at all. But if you guys have any questions, I'd be happy to address those.
Amazing. Thank you so much, Patrick. Cool. So I have some questions here for you. I'll try to keep them a little bit organized so we're not jumping too far all over the place. So I guess the first kind of group we'll do is on the cameras and trackers, kind of the equipment that you guys are using. So is I guess first of all, what's the kind of cost per unit of that? Like if you're tracking one seal, or whether it's via camera or just kind of GPS tracker, what's the price there?
Yeah, so the instrumentation cost is super variable, and it just depends on the exact study goals. So I'll start off with like kind of the simplest and then work up to the more complex. So If we want to put a time depth recorder and a simple satellite tag on an animal, we're talking about $3,500 per animal of equipment. And then on top of that, the Argos satellite tracking, we currently get that covered for free by the Animal Tracking Network. That probably is not going to last forever, but functionally that usually costs about $1,000 per year per animal. So call it $4,500 for a year of tracking. So that's on the lower end of the spectrum. And then these kind of fancier tags that record conductivity, temperature, depth, or other more advanced features like that are upwards of like $7,000. And then these super fancy custom instruments, it's almost like impossible to put a price tag on them because they're not commercially available. So it's just whatever it costs the researchers to work with manufacturers to create them. But some of the contracts that they have, if I had to put an estimate on it, like $15,000 per instrument, something like that. So it's pretty expensive. Dan Costa and Roxanne Beltran have tried very hard for upwards of 20 years now to have a continuous stream of funding to support this work, and it's hard for them. So not too many people can pull that off. Yeah, it requires a lot of money to keep that project going.
Awesome. Yeah, makes sense. And then it seemed like most of the studies that you were doing were on females. Do you also tag males ever, or are females just like kind of more interesting for your purposes given the fertility association and everything?
Yeah, so over the years we have put instruments on some males. In general, the main reason we don't is because from a demographic perspective, they're a little bit less interesting, but also they have a higher mortality rate. So if we're putting out 20 sets of instruments, we kind of want to get most of them back because we get a full archive of data and we can reuse the instruments. And at those costs, it's really nice to be able to reuse a bunch of the instruments each time. For the adult females, we have about an 85 to 90% recovery rate, which is just unheard of in terms of the biologging community. Like if you put out, 100 tags in tuna, you're very lucky to get back 15 of them. So we're very fortunate on that front. But yeah, in terms of the males, there was one, there's been a couple of projects for the past 20 years, but I think the total number that have been instrumented, is on the order of maybe like 30 or 40. So yeah, very small number. I should point out that Professor Beltran right now is putting out instruments on an equal number of male and female juveniles. So I mentioned those different foraging strategies. It was not clear when those differences developed. And so she's very interested in looking at that. So she has weanlings, yearlings, 2-year-olds and 3-year-olds, half male, half female that she's instrumenting right now. And this is the final year of her project. So stay tuned on that one. There'll be a publication out in the next few years about that.
Awesome. Well, super helpful. Thank you. And then as far as I guess next kind of broader category is I guess the Nuevo population specifically. I think you mentioned I guess it was towards the beginning that they started showing up in the 60s. Is there a theory as to why then?
Yeah, so I should say it's impossible to know for sure. So the formation of new colonies is sort of a mysterious process that we don't fully understand. Folks have done analyses that look at beach topography and offshore rocky reefs and things like that to know like which areas might be good habitat for them. But they don't always choose the best habitat for whatever reasons. So it's really hard to know. Functionally, like from a larger perspective, the elephant seal population was growing exponentially. So that it sort of like filled up all of the space in Southern California on the Channel Islands and then needed to expand somewhere. And so, it's probably the juveniles that first explore areas and then the male, and then finally a female shows up and gives birth to a single pup. And then, maybe nothing happens, but maybe other females are attracted to that. And then those pups, tend to return as adults back to the same area where they were born. So then you get a colony formed. It's worth noting that there aren't very many islands along the Northern California coast. So, Nuevo is probably one of the only places north of the Channel Islands where they could reproduce. So that was probably kind of like the first opportunity. And once they grew large enough in their population numbers on the island, so like the main beach out there was completely full, then they sort of spilled over to the mainland. And now there's more seals breeding on the mainland than there are on the island. And then subsequent to all of that, they started breeding on the mainland down at Piedras Blancas, and then they've been expanding northward as well. So my kind of guess on all of that is just that they were expanding northward, and that was the first place that they found that was an island.
Makes sense. And then you mentioned their kind of feeding patterns along like the gyre boundary. What's kind of the reason for that area being so fertile? Like, what's the is there kind of a some sort of current pushing something up, or what's the geographical explanation there?
Yeah, so it's upwelling. So it's not like the coastal California upwelling that's super, super strong, and we see these giant blooms that happen. So it's not quite that large in terms of its magnitude, but it's large in terms of its geographic area. So it is an amazing amount of resources that are being pumped up that get to shallow water and then feed phytoplankton and then on up the food web. So there's that nice balance there between the oligotrophic waters to the south and the highly nutrient waters to the north that are colder and then mixing in that area. So yeah, it's just, and that shifts quite a bit depending on the season. So I didn't show a plot of this, but the oceanographers like to talk about the TZCF, the transition zone chlorophyll front. So on an annual basis, because of the seasonality, that there's a line basically that forms of chlorophyll, so phytoplankton, that marches south, as the summer progresses and then retreats back north during the winter. And the elephant seals, are sort of focused in on that general area. So it's a very dynamic area. It's not like in the center of the gyre, there's just less going on than at that boundary where there's more upwelling.
Makes sense. And then towards the beginning, you had that great chart with I think it was like 80-something% of females that never reproduce. In the teens percent that reproduce, have you noticed, or have there been kind of studies on commonalities of those females? Like, are they better hunters? Are they bigger? Like, is there some sort of success factor?
So you're saying that of the ones that are super successful, is there a pattern there?
Or successful at all?
Yeah, so the ones that, so the age is the main factor there.
Got it.
So oftentimes, like once a seal reaches age, 4 to 5, they're very likely to survive and reproduce after that. So there's a higher mortality earlier on. So really a lot of that difference, the reason that they don't make it to reproductive age is because they just don't survive those first few years. And so Professor Beltran is working on that question right now. So stay tuned on that one. We haven't tracked very many of the juveniles until just recently on the older age. So the ones that make it past like 10 years, and have up to 10 or more pups. Yeah, we have examples of individuals that behave like average seals. We have examples of ones that are sort of unique. I think our deepest diving seal was one of the oldest as well. I happen to know her number, so it's 1885 is her flipper tag number. She was the one who was the deepest diving seal. So we tagged her a couple of different times. So she broke the record on her first migration, and then we instrumented her again on a subsequent year, and she broke her own record. And she decided to feed up off like kind of close to Vancouver Island on the continental shelf break. So I would love to be able to say like, oh yeah, the ones that live a long time are the ones that stay away from the coast where it's safer. But one of our oldest, most productive super moms was one that fed kind of close to the coast on the continental shelf there. So I'm not sure we have enough of a sample of the really old productive animals to be able to make any general patterns like that. All I can say is they're really good at not dying.
Makes sense. Well, cool. Now we have a couple on more the conservation side. So I guess the first is what's kind of your and the community's biggest man-made conservation concern? Like, is it underwater mining or ship strikes, or what's the what keeps you up at night, I guess?
Yeah, this is sort of a weird answer, I realize, because it's not a threat yet. But I mean, in general, elephant seals are doing fine. They're in the least concern category of the IUCN list. So by all accounts, they have a very large population size that is continuing to grow quite rapidly, perhaps beyond their historic numbers. So there's certainly not a major conservation problem with elephant seals. But that being said, one thing that does worry me is the possible expansion of fisheries into the mesopelagic zone. So, humans are very good at exploiting natural resources, and as of yet, no one has figured out, or no company has figured out a way to exploit the myctophid fish resource. And it turns out that that is one of the most rich fish resources on the planet in terms of biomass. So it would certainly be very lucrative if someone figured out how to do that. I think that they might not find it lucrative because they're not schooling in the same way that krill or anchovies, sardines typically do. So I don't know how they could do it efficiently. But if someone figured that out, then that could potentially have a huge impact, not just on elephant seals, but on all of the other species that rely upon that pretty amazing resource out there. So there's scientific papers and people exploring the possibility of myctophid fisheries. And I don't know, if I had to zoom ahead in 100 years, I would be very fearful that we were exploiting that resource and not just letting the seals take care of it.
Makes sense. And then there was one question on, do you know anything about, someone said that there might be a declining population on the Farallones. Have you heard about that, or if there's anything specifically causing that?
Yeah, so I don't know specifically what would be causing that. In short, the Farallon population is very small, and that's not great habitat for them, and it's the very edge of their range. So what we might be seeing there is just sort of a random effect there of the conditions, and not an indication of the entire species. So this is just me guessing, so I don't know for sure, but that area is very rocky. Elephant seals do much better in kind of open expanse sandy beaches. And so a combination of the habitat and being on the edge of the range there probably just means that individuals might prefer other areas. So if I had to venture a guess, I would say those females might be slowly emigrating elsewhere. But yeah, certainly not an indication of the health of the overall population. Got it.
Makes sense. And then last one on I guess that side is on the predator front. So what are their biggest predators, and kind of how big of a concern are large predators? Like what percentage of elephant seal deaths are caused by predation?
Yeah, so it's a really hard thing to get at. Predation out in the open ocean, like away from where we can see it, is just a really hard thing. So probably, we have records from many animals that have been eaten by other animals, but the problem is we don't get a record of that. We just see our nice satellite track blink offline, and we never see the animal again. And we don't know if it got struck by a ship, if it starved, or if it got eaten by something. The one exception to that is Professor Beltran right now in that juvenile study that I mentioned. So she calls that study, it's kind of a horrible name, she calls it Rules of Death, because the idea is to study what is going on, like why the juveniles have a higher mortality rate. So she's putting out these tags that give us a little bit of a window into at least predation versus starvation. And so she's able to do that by using some pretty fancy tags that were specifically developed for southern elephant seals, actually. And then she was using them for northerns. And they have an acceleration logger built into them. And it's kind of tuned to get at the head lunges. So similar to the jaw motion loggers, just whenever they lunge forward to capture prey item. But the difference is you don't need to get the tag back. So it processes those data on board, sends it via satellite, and then we can just look through the record. Even if the animal doesn't come back, we can see how well it was doing. And so if it was feeding a lot and then just blinked offline, probably that was predation. If it was not feeding very much at all, and the condition was getting worse and worse, feeding less and less, and then blinked offline, maybe that's starvation. So we're trying to get at that difference now, but it's a pretty hard one to assess. As far as their predators, we think that white sharks and orcas are their main predators. But again, it's really hard to know. Like we don't have very many direct observations. I certainly don't. I've been studying these seals for 20 years. I've never seen one be eaten. I've seen evidence of shark bites, giant wounds in them. So we know that that's happening near Año Nuevo, but as far as what's happening way out at sea, it's really hard to know. But hopefully this study will give us a little window into that.
Makes sense. Well, I think that's it from the Q&A front. Thank you. I know that was a lot of rapid fire, but really appreciate it.
Yeah, no worries. Happy to.
Yeah, I just want to say, it sounds like we have to have Dr. Beltran come and speak.
Yeah, I would highly recommend that.
Maybe you know how we could get in touch with her.
I can send you her email, yeah.
Anyway, yeah, well, it sounds like quite a power couple. And yeah, it's been so fascinating. I think I'm like many of us tonight, will never look at the elephant seals kind of lazing around on the beaches with anything other than admiration, and that the fact they well deserve their rest time. It's just amazing.
Yeah, we joke that these guys are just amazing athletes, and we're just seeing their like post-athletic vacations on the beach.
Yeah, and just the evolution to be so adapted. Yeah, no safety stops.
Nope.
Well, again, thank you so much, Patrick. It's really just been fascinating, and yeah, it's good to know all that's happening, all the research, and all we still have to know and discover.
Yeah, of course. And I forgot to mention, but if folks are interested in kind of following along on the Beltran Lab website, there is a map that contains the live tracking data from the seals that are out at sea right now. So she's just deploying instruments right now, and so you'll be able to follow along with those for the next like 7 months until the animals come back again. So that'll be fun for everyone.
So we just do a search for Beltran Labs. Yeah, how do we get there?
Yeah, so if you just search for Beltran Lab at UCSC,
Yeah.
I can probably give you the exact URL here.
Or if you have it, you could just put it in the chat. We'll capture it, because I bet there's there'd be interest in that. That's great. The one thing
Yeah,
while
there we
you're
go.
doing that, that some of those devices looked really big. Is there any issue with, I guess, there's no way of really knowing that whether the elephant seal is uncomfortable with that, but
Yeah, so we get this question a lot, and so I like to mention a few different things. So one is that the seals are fully capable of using their front flippers to reach the top of their head or their back. So, if they wanted to try to rip off these instruments, they certainly could, and they don't. So they're not immediately discomforting for them.
yeah.
So that makes me feel a little bit better. But then also we can do studies based on the flipper tags, and basically, if we have a large number of animals that are carrying instruments, we also have a much larger number of animals that are not carrying instruments, but that we still know a lot about in terms of their, the demography and everything we can learn from having flipper tags in them. So we can look at things like their mass gain with and without instruments, how often they give birth with and without instruments. So basically all these different metrics that we can compare. This is going to sound weird, but when this analysis was last done, the animals that carried instruments actually did a little bit better than the control group. And we think what's going on is that we choose healthy animals to deploy instruments on, like we're not going to choose the animal that looks sick. So that probably biases things a little bit, but at least we know that we're not causing a big negative detrimental effect to them.
Yeah.
And then there have been some studies on the drag associated with tags. And some of the tags, like when I compare it to my body size, it's kind of large, but elephant seals are pretty massive animals, and the speed with which they go matters a lot. So if you put the same tag on a sea lion that's of similar size, that would matter a lot more because the drag coefficient goes up exponentially with speed. And elephant seals are just traveling at a slower speed. So we think they can just tolerate a larger instrument without causing them much additional energy expenditure.
Wow. Yeah.
So in many respects, they're just kind of like the perfect research partner because they can tolerate all this stuff. They're easily accessible, doesn't impact them very much. So they can tell us a lot without being detrimental to them at all.
Wow, fascinating. Yeah, well, I think we'll have to have you back too, Patrick, if you will grace us. It's been great. So again, thank you so much, and thanks everyone for attending, and we'll see you again.
Yeah, thanks for all the great questions. Bye-bye.
Bye-bye.